Welcome back, ladies and gentlemen. So before break, we talked about protein folding, specifically the different stages of protein folding. The first stage being your primary structure, which included your beads on a chain, your beads being your amino acids. So it looks like almost like a pearl necklace in this representation here. But what this actually is showing the primary structure of a protein. So this is where you actually have the different orders of amino acids in the molecule itself. So when the amino acids line up in different orders, they have different attractions to other amino acids in the chain. What this causes is the secondary structure. So this is the primary, even though it's called the secondary structure, it's the primary folding type in the actual protein. So here you have a beta-pleated sheet, they call it, which it kind of looks like a bunch of sheets of paper in this representation. And it's basically a linear arrangement of amino acids back and forth. The other type of secondary structure is called an alpha helix, and they call it a helix because if you notice, it looks sort of like a DNA helix, except it's not double-stranded. So this is an alpha helix. And what ends up happening is these two secondary structures end up folding on top of each other to create a tertiary structure. So this globular-like structure, or what they often call a globular protein, is the actual tertiary structure of the amino acid chain you see here. And your different side units, or your alpha helices and beta sheets, will have attractions to each other. And again, this is based on the difference in the amino acid configuration or sequence. Now, proteins can exist and function at the tertiary structure. However, some proteins form complexes called quaternary structures, which include multiple subunits, so multiple amino acid chains put together. So if you notice, this one individual amino acid chain folded on top of itself and created one subunit, one tertiary structure. Other amino acid chains separately create different structures and then combine to make this one quaternary structure, which can have a completely separate function when compared to its tertiary structure components. So this should be a review, but this is important for what we're going to be talking about today because if you remember, each protein has its own separate function, and since enzymes are proteins, they have their own specific functions. And that function is determined by the shape that is determined by the amino acid sequence, causing different folding patterns in the actual molecules. So one thing that ends up happening and can affect enzyme reactions is temperature and pH. Before break, we talked about this very briefly, and it was on your quiz, but I'd like to talk about it again because it's important for understanding how different conditions can affect enzymes and the functioning of enzymes. So if we look, proteins work best under specific temperatures and pH levels. Let me give you an example, for instance. Our proteins in our body work best under our average body temperature of 98.6 degrees Fahrenheit. If we have anything above or below 98.6 degrees Fahrenheit, we are outside of the optimum or ideal range of temperature. Our pH in our body is right around neutral, so anything more acidic or more basic than our normal body conditions would also have an effect on the folding of proteins. If either the temperature or the pH changed, as indicated above, either going above or below the norm, the protein begins to unfold because the interactions between your secondary and tertiary or your secondary structures, such as these helices and the sheets here, they start to break. As they break, the protein starts to unfold, and this is called denaturation. Okay, so the change in shape of an enzyme or protein is called denaturing, which can severely affect the protein's function. Okay, so denaturation you end up basically putting the protein back into its primary structure, or at least unfolding it to the point where its shape has changed enough so that it does not perform its function. Now, proteins can regain their shape if you go back to the normal temperature. However, some proteins can be cooked, we'll use as a good word because we'll talk about it in a second, can be cooked to the point where they will never go back to their conformation. So some proteins can go back to their conformation, some proteins can't. So let's watch a small little video about protein denaturation, and you'll get a better understanding of what actually happens. So if you look here, you have a protein that is in its tertiary structure, either secondary or tertiary. And you see these green bonds that are between the different side chains of the protein itself. As it heats up, those bonds break. Okay, so you see the breaking of the bonds here. And when these bonds break, the interactions between the side chains of the proteins start to unfold, and you end up having this long protein chain that is unfolded and essentially has no function. So we all probably at one point or another have seen or eaten or even cooked eggs. So this is actually a great way of talking about protein denaturation and how it changes the actual physical nature of the proteins. So if we go back to our little PowerPoint here, and we take a look at eggs, okay? If you look here at the egg on the left, this is probably a common sight if you ever bake or cook or anything. When you first break an egg open, you see this translucent or almost see-through part of the egg. This is all protein. Okay, so eggs are high in protein, and this is where a lot of the protein in the egg is found. Now, this protein, if you notice, if you look here, is folded in its tertiary and quaternary structures. You see the secondary structures here with the helices, and you got some sheets in here. But what these do is, when they're folded up, their shape prevents the proteins from being very closely associated with each other, leaving these gaps. And I'm going to circle them. You see these gaps inside the protein? These gaps hold water, and so the reason why the inside of an egg is liquid, all right, or in the cytoplasm of a cell, the reason it's liquid is because it holds water in the gaps between the proteins. So the cytoplasm here in your egg is actually a combination of water and protein. Now, when you fry an egg in a fry pan or however you cook your eggs, what ends up happening is your proteins denature and they unfold. So you get these long chains. When these long chains unfold, you actually allow the protein to become closer together. When that happens, water is kicked out of the spaces in between the proteins. Okay, so it's almost dehydrated. So the protein in the egg is dehydrated. That's why you see a lot of steam coming off the egg when you cook it. And the water comes out. When that happens, you no longer have a liquid, you actually have a solid, and that's what actually happens when you cook an egg. So denaturization or denaturing is an important aspect of cooking, but it's also important in our bodies. If you think about it, would you want this to happen to your proteins inside your body if you had too high of a fever? Of course not. So this would be very detrimental and cause instant death if this happened in your body. So that is why our body needs to be regulated and cannot go above temperatures such as like 105 and 106 degrees. Because if that happened, our proteins would denature to the point where they become unfunctional, specifically our enzymes, and our reactions would not be able to occur. So this is a short little reminder of how protein denaturization or denaturing causes changes in proteins and enzymes that result in functional changes in those proteins and enzymes.